everyone. So welcome back. Today I'm going to start my second topic, and today I've selected cellular respiration. And so let's start with cellular respiration. First, I'll tell you what it is, and then we will move on to our topic of Krebs cycle. As I mentioned earlier, today we'll be talking about Krebs cycle. So first of all, what is cellular respiration? So you know that we all consume food. Why do we consume food? Do you know that? Because so that we can obtain energy from the food. So how do we obtain energy from the food? How does it occur? So when we take in oxygen, then this oxygen is used by inside our body. So it is used to break down all the food that we're taking. That is the carbohydrates, proteins, amino acids. So when these are all broken down, the nutrients are broken down to release energy. This process is known as respiration, and when it is using oxygen, it's known as aerobic respiration, and in absence of oxygen, it's known as anaerobic respiration. So today we'll be talking about aerobic respiration. So in this aerobic respiration, there are four steps that are occurring. So first step is glycolysis, wherein the glucose is broken down to form pyruvate molecule, and then we come to the next step, that is when acetyl CoA is formed from pyruvate by oxidative decarboxylation method. And the third process is out here is Krebs cycle. And this Krebs cycle, what happens out here is this acetyl CoA is utilized, and energy is produced in the form of ATP, NADH, and FADH2. So, and the last step is electron transport chain, wherein oxidative phosphorylation occurs, where this electron carriers that are produced during Krebs cycle, they are transferred to they transfer the electrons to electron transport system, wherein the rest the entire glucose is oxidized. So the last step actually, wherein energy is finally produced in the form of ATP. So today I'll be talking about Krebs cycle. So how did this Krebs cycle name come from? Like, where did it come from? It came from a person's name known as Hans Krebs. So Hans Krebs in 1937 he identified this Krebs cycle, due to which he also got Nobel Prize in Physiology or Medicine in 1953. So this Krebs cycle, it is basically a biochemical process wherein amino acids, protein or glucose, these are all broken down to form products such as carbon dioxide, NADH, FADH2, ATP. These are all forms of energy. So this process is also known as amphibolic process, because in this cycle both the anabolic reactions and also catabolic reactions both are occurring. So this is also known as amphibolic pathway. So let's move on to this Krebs cycle. So later I will tell you the importance of the cycle, and let's move on to the diagrammatic explanation. So here I'm going to tell you that how the process is occurring, the cycle of Krebs cycle. So basically, Krebs cycle consists of 8 steps. Okay. So the first step out here is when glucose forms pyruvate and pyruvate forms acetyl CoA. The acetyl CoA is utilized in this process. I'll just okay. So there is a way of remembering the cycle. I'll tell you the way later. So okay, fine. I'll tell you right now only. So I have like created one sentence by which we can. It's like a trick type by which you can remember the entire process. Okay, by which we can remember the names of the molecules and compounds that are taking part in this Krebs cycle. So the sentence is: on a calm illuminated night, a kind noble saint freed a furious melodious nightingale. I know it's a bit tough, but I think you can memorize this, right? So on a calm illuminated night. So first O, right? So O out here is oxaloacetate. So on a calm. So here this is A, okay? Just a minute. Okay. So this is out here A. So OA. C is out here citrate on a calm. Illuminated I. This is iso citrate. Okay, on a calm illuminated night. So out here night I have shown as NAD to NADH. This is one product out here. So on a calm illuminated night, a kind noble saint. A kind noble saint. So out here A stands for alpha ketoglutarate. A kind, okay, so AK out here, alpha ketoglutarate, AK, a kind noble saint. So noble out here again N stands for NAD to NADH. So here first succinyl CoA is formed and then succinate. So out here it stands for S, okay, this saint. Freed, a furious melodious nightingale. So freed, that is F, right? So F I have kept here as FAD to FADH 2. Okay, freed A, right? So A again ATP. Okay, freed A, furious. So furious stands for fumarate. Then melodious, malate. Nightingale. So again nightingale stands here for NAD to NADH. Okay, so here this is the sequence. So on a calm illuminated night, a kind noble saint freed a furious melodious nightingale. Okay, so this is a sequence which you can remember, which you can use. Okay, so okay, we'll come back to this one only. So right now this is the sequence. Okay, now we'll talk about what is happening out here. So oxaloacetate out here, it's a 4-carbon compound. Okay, so then when it's forming, acetyl CoA is utilized here and oxaloacetate is using it to form citrate. Okay, so CoA, right, out here this coenzyme A is released and we are getting citrate, which is a 6-carbon compound. Okay, so this is occurring with the help of the enzyme citrate synthase. This is the first step. Then from citrate we are getting isocitrate with the help of the enzyme acetonitase. This is the second step. Okay, third step is when isocitrate is forming alpha ketoglutarate, and out here what's happening is dehydrogenation is occurring. So here it's occurring with the help of the enzyme isocitrate dehydrogenase. Okay, just remember this process. Okay, dehydrogenation. So here you can understand it's dehydrogenase, sorry. Okay, this is out here, this is the third step, and during this step carbon dioxide is released. Okay, now the next step is alpha ketoglutarate. Even out here same thing, here it's using alpha ketoglutarate dehydrogenase. So I'll just a short form, dehydrogenase. This is the fourth step that's occurring. Okay, this is forming succinyl CoA. Okay, so after that, during this process again, carbon dioxide is released. Okay, I'll write it. Okay, so now from succinyl CoA, succinate is formed, which is a 4-carbon compound. In some books you will see that they have shown directly from alpha ketoglutarate to succinate, but if you look at the detailed, then there is also another step out here. This is the 4th step, and from succinyl CoA to succinate is the 5th step actually. Okay, so this 5th step is I'll just write it down here, 5th step. Okay, so during this step an important thing is happening, that is the formation of ATP. In some cells GTP is formed. Okay, so this is occurring with the help of succinyl CoA synthetase. Okay, synthetase. Fine. So again from succinate, fumarate is forming, which is again a 4-carbon compound. So this is being oxidized to fumarate. Out here the enzyme that is used is succinate dehydrogenase. Okay, so this is the 6th step. Then again this fumarate is oxidized to malate, which is again a 4-carbon compound. Using the enzyme that is helping in the process is fumarase. During this step water is being utilized. Okay, and the last step again out here is when from malate oxaloacetate is being formed. So this fumarase is 7th step. This is the 8th step, wherein malate dehydrogenase is used. Malate dehydrogenase. Okay, this is being used. So the cycle is repeating again. Okay, so the cycle occurs twice. Why does the cycle occur twice? Because here 2 molecules of acetyl CoA is produced from 2 molecules of pyruvate. Therefore this cycle is taking place 2 times. Fine. So now we talk about the products that are being formed. You can see out here NADH, right? 1, 2 and 3. You can see out here FAD out here once, right? FADH2 is formed once. Then you can see carbon dioxide 2, right? Here and here 2. Okay, ATP once. So let's go to the next page. I'll just take you to the next page. So here I'm going to write the products of Krebs cycle. Please remember, this is very important for medical examination and also for students who are in class 11. So this is important, you should know these things. So what are the products out here? The products out here is 2 carbon dioxide, 3 NADH, okay, 1 molecule of FADH. So I'll just write 1 FADH2. Okay, so 1 molecule of ATP or GTP. These are the products. Okay, so since out here 2 cycle is occurring, therefore the products will be how many? 4 CO2, 6 NADH, 2 FADH2 and 2 molecules of ATP. So this is one. So now a very important question out here is how many molecules of ATP is generated in this process. So if I talk only about Krebs cycle, then it's 24. How it is 24? Let's talk here. So see out here 6 NADH. As you know, one molecule of NADH will give 3 molecules of ATP, right? So there is 6 out here. Therefore this is 6 NADH will give us 18 molecules of ATP. 1 FADH2 molecule gives us 2 molecules of ATP. Therefore, 2 FADH2 will give us 4 ATP. Okay, and then outer ATP is how many? 2. So total is 24. So in Krebs cycle is 24, but if we talk about cellular respiration, in cellular respiration this is 38 molecules of ATP. Very important, remember this. Actually it's 36, but 2 ATP is formed outside the mitochondria, so along with that it comes around 38. Okay, so now the next, let's go to the next page. So Krebs cycle, where does this Krebs cycle occur? Krebs cycle occurs in mitochondrial matrix, but this is in case of eukaryotes. So if we're talking about prokaryotes, then this process is occurring in cytosol. Okay, so the energy that is being this electron carriers, that is the NADH, the FADH2, this ATP, these are all being carried on to the next step, that is the electron transport chain. Okay, wherein oxidative phosphorylation is occurring to form energy. Fine. So this is the diagrammatic explanation of Krebs cycle. Okay, so I hope you have understood the process. So just last we'll just summarize the process with a formula, that is the acetyl CoA plus 3 NAD plus 1 FAD plus 1 ADP plus 1 phosphate will give 2 molecules of carbon dioxide, 3 NADH, 3 hydrogen plus 1 FADH2 and 1 ATP. Okay, so out here, since like this is the entire process of Krebs cycle, okay, you should remember this. So now let's go back to our video. Okay, so I hope you have understood Krebs cycle. So why is this Krebs cycle important? This Krebs cycle is important because what does it give us? Energy, right? So from Krebs cycle we get a lot of energy, and without energy can we do our daily activities? No. So like when we take in oxygen, why do we take in oxygen? Because this oxygen is used to break down the food materials and derive energy. Okay, this is very important. And also it's important because the products that are being formed, okay, during the Krebs cycle, like NADH, FADH2, these are all electron carriers, and they are helping in further process of cellular respiration, that is they move on to the electron transport chain and form the energy. So Krebs cycle is very, very important, and it's not only important for knowledge, but it's also important for your exam. It's a very important question. And one fun fact or information I would like to give you as usual. So let me, okay, can I tell you? Okay, so stop the video. So do you know that the athletes, okay, the athletes, since we're talking about Krebs cycle and it's occurring in mitochondria, the mitochondrial matrix, mitochondria, so I'll give you an information of mitochondria. So in athletes, like if you have seen the structure of mitochondria, there's a part known as Christie, right? So there's a modification in Christie, okay? That part is there's a modification in this structure of the mitochondria in athletes, because it helps them, because since they are doing a lot of physical exercise, so it is helping them to the muscles to adjust to the exercise and to the pressure of the exercise. So this is one information I gave you today. So we'll meet again in next class. I hope you have liked it, like today's class. Thank you so much.